Hi, everyone, and thanks for joining us for our discussion of immunotoxicity, COVID-19, and CRS today. My name is Aaron Rose, and I'm a program manager with the in vivo team at the Jackson Laboratory Facility in Sacramento. My background is in oncology and immunology. In the Sacramento group, we do a lot of work with immuno-oncology models and mouse humanization overall. I'm here today with Jim Keck, and Jim is the head of our product development team. This team largely focuses on internal projects and collaborations with outside groups intended to create new experimental models to address the scientific needs of an ever-changing drug development landscape. Given that introduction, Jim, I think I'd like to kind of start things off with a really general question, and that is, can you tell me what is CRS?
Cytokine release syndrome, CRS. It's I would say, a outcome of overactivation of the innate or various parts of the immune system, which leads to high levels of cytokine release, leading to downstream effects on the body, organ failure, fever. It is definitely a downstream event that's leading to toxicity that's not desired. Okay, when it comes to therapies or severe infections.
Now, neither of us here are clinicians, but today we are talking about coronavirus and COVID-19. We follow the news, we're following the clinical trials and the newest data that's coming out. Given the caveat, can you tell us a little bit about how CRS might be associated with COVID-19?
Well, CRS is one of the major downstream events that happens with severe COVID infection. A lot of the patients you're seeing that are hospitalized or actually unfortunately passing away is related to cytokine release syndrome, high levels of cytokines. It's involved in the immune system trying to tackle the virus or the virus itself causing cytokine release. It still needs to be studied a lot deeper. As you know, we're only a couple of months into the infection. People are still trying to understand the implications of the CRS and how it is induced. But it's a severe issue when it comes to COVID.
Now, CRS as a sort of topic has been bubbling to the surface more and more as we start to explore biologic and cellular-based therapies. Given that sort of history, can I ask you how the CRS platform developed, where the idea came from, and how you built it up in your lab?
Sure, I'll be glad to answer that, Aaron. So we've been working in CRS for over 4 years. What happened was about 5 or 6 years ago, we started getting involved in humanized mice for immuno-oncology. It's a relatively new platform, especially then, but still now. And the idea was we were looking at a humanized mouse, usually CD34 or PBMC humanized mouse. You put in a patient-derived xenograft for a cancer cell line, and you look at various drugs for activating the immune system to kill the cancer. That was started with checkpoint inhibitors, but as we were moving into CAR T and to bispecifics, we started seeing downstream toxicities that weren't predicted. What happened was that we started seeing the mice temperatures dropping. We see severe cytokine release and death. And so we started to investigate what's going on here. And the more we dug into it, the more we realized that cytokine release syndrome is a significant issue when it comes to immune therapies, especially with CAR T. So we started to develop a platform that we wanted to make sure that it was sensitive, reproducible, and reliable. And we wanted to be able to do it in I want to say almost like a personalized medicine type approach. We didn't want to ask questions of yes or no. We wanted to know in a population of 3, 5, 10, 50 people, okay, who would get CRS with the therapy versus somebody else who wouldn't get it, and why? And that's how we got started in this whole platform.
I think that's a really exciting point, and I'd like to sort of grab onto that idea. I know that for a lot of us who are involved in the development of biological therapeutics, stories like what happened with Tagrisso it wasn't Tagrisso, I apologize, T. Gennaro, really stressed out the the clinicians who are developing these biological therapeutics. And my question for you is, given that, are clinicians now reaching out to you to see if your CRS platform might be used to help them screen for the clinical trials or pre-screen before even going into clinical trials?
Sure. So our assay actually picks up T-Genero 1412 analog. It's essentially the same amino acid sequence as the drug itself that caused such significant issues in phase 1 in 2006. So our assay is sensitive enough to pick up a drug that was missed during the primary screens in 2006. So people are interested in that our platform can pick up that agent as well as other type of agents that are toxic in the clinic. And people are starting asking questions about, okay, how can I utilize this platform for the clinic as well as basically preclinical studies? And one of the things we're doing right now is we're looking at efficacy and toxicity for immunotherapy, where we basically put a tumor into the humanized mouse, and we look at the activation of the immune system against the tumor, but also the toxicity, and we correlate different levels of antibody or therapy to find which dose is appropriate for efficacy with minimal toxicity. In the case of clinicians, people are starting asking questions about, could this assay, because it's a PBMC platform mostly, you ask questions about what is the clinical correlation with this assay to what's happening in the clinic? Because right now there's no reliable assay or a preclinical assay that's been validated that correlates what's going on in the clinic. So people are looking at this platform as a possibility for asking those questions because we use adult PBMCs, and we can actually look at individual responses.
So a lot of concerns in the early days in the development of drugs to downregulate the immune system was that you'd be opening yourself up to infections while treating autoimmune diseases. Is it possible that the converse is true? That is, if we are augmenting immune cell function, we may be inviting CRS in? And if so, I'm asking because I've seen this in some of my own experimentation where sometimes the greatest efficacy I see in a particular, usually in CD34 donor humanized mice, sometimes the best efficacy I see is in the mice that lose weight and look kind of unhealthy, and I suspect that there may be some element of CRS or an overactive immune system there that's causing that mouse to be able to take out that tumor. Do you see that in your combination CRS efficacy experimentation?
Absolutely. Okay. Sometimes the most powerful drugs that cause complete tumor regression or elimination, okay, are the ones most toxic. And so that's what the problem is. You need to be able to find an efficacious dose that minimizes toxicity, because if the patient can't handle the drug, you're not going to get the tumor shrinkage. And it is a significant deal. And so for this, we're talking about the activation of CRS by various drugs. The fact that we can actually shift this platform and also now look at COVID-19, where patients are getting CRS or caused by the virus itself without any therapy being involved, as the virus-induced disease. Our platform can be leveraged to be asked questions about CRS in the context of the potential virus infection. So a lot of the cytokines we measure and the outcomes we look at with immune therapy, we do the same thing for COVID-19. To give you an example, IL-6 is really high with our positive controls in our assay, and IL-6 correlates with poor clinical outcome with COVID patients. Okay, so we measure that. We measure IL-8. We can look at IL-1 beta. Now, in the clinic with COVID-19, it's mostly an IL-6 phenomenon, less of a IL-1 beta phenomenon. But it's surprising because macrophages are supposed to be involved, and macrophages are involved in using both cytokines. Our platform, our PBMC platform, picks up IL-6 readily, as well as IL-8, GM-CSF, and other cytokines. And it's in some ways it mimics what we're seeing with the COVID infections in people.
Excellent. I'm going to pause for a second and acknowledge that we've got some visitors with us today from Austria, India, Bangladesh, Texas, California, and Maine. And I'm going to go ahead and encourage all of our audience members to submit any of their questions in the comment area today. We'd love to address any of those questions that we can while we're on here with you. So thank you for joining us. And I'll jump back in. We were briefly discussing a little bit there, Jim, about donors. And this is something that comes up all the time when I'm having conversations with people about setting up humanized immuno-oncology experiments, the donor-to-donor variability. You mentioned the fact that at the end of the day, you'd like to be looking at a population of people to understand potential for development of CRS given therapeutics. Is this something that you're currently exploring? Are you having those conversations with researchers coming to you, and are they starting to use multiple donors in these experiments?
Sure. So let me address this, Aaron, your question in two different ways. Donor variability, but also reliability. If we do the same PBMC donor 5 times, we get the same result 5 times in 5 different experiments. We get a highly reliable assay for looking at one individual with the same therapies over multiple times. So it's a reliable assay. But in that context, you not take 10 different people and put them in your assay and look at your therapeutics. In those 10, you'll see 10 different cytokine responses to a therapy. Some are similar to others, but not some are completely different. So the significant donor variability, which is what you see in the clinic. Not everybody gets to toxicity with the therapy at the same time as somebody, or at the same dose as somebody else does. So once again, we get reproducible patient response to a therapy, but there's differences between you and I and how we would respond to 10 milligrams of some drug. And that's important for us to know, because as you're going into clinical trials, you want to know, what is my phase 1 going to look like, essentially? If I look at 25 different healthy PBMC donors, and I get an array of responses, it might help me predict what I'm going to do in phase 1.
Can I ask what kind of therapies people are asking you to test in this platform? Is it limited to just checkpoint inhibitors?
No, we're doing a lot of work with bispecifics and CAR T. And I also would say that we've got a lot of data looking at a combination therapy, bispecifics with so experimental bispecifics with clinically FDA-approved drugs in combination. And in combinations, we're seeing, once again, donor variability and how responses. And we're finding some surprising synergistic toxicities in some individuals that I would never have predicted going forward. Another thing we're starting to see is mechanistically different. 2 different drugs combined together have 2 different targets, leading to additive or enhanced or even synergistic toxicities. So we're asking a lot of those kind of questions as well. So as your question, CAR T, bispecifics, things like that are mostly what we're doing right now. Less with checkpoint inhibitors because they don't cause a lot of CRS in the clinic.
Excellent. So I mentioned at the top of the conversation that I work with the commercial group, and that you head up the research and development group, our product development group. I'm wondering, so your group has developed CRS. Is this readily available? Are we already offering this as a product for folks who are out there listening and thinking that this might be something of use to them?
Sure. So a couple of things. We work very closely with the commercial group, research group. We are doing the CRS assay at least 3 times a week. So we're doing this assay a lot, okay? And we're doing it for our own research, but also for people who like to test their drugs in a platform and asking questions about efficacy and toxicity together, or just toxicity. But we're also starting to looking at inhibitors of CRS related to COVID. So we're seeing a lot of people interested in finding new ways to treat CRS in a clinical setting. So once again, a therapy that inhibits CRS for COVID might be a good therapy to treat for CRS when it comes to bispecifics or CAR T as well. So I would say it's blossoming. A lot of people are trying a lot of new ideas and nice things to try. So it is robust from that point of view. So yeah, it's something we're doing not daily, but darn close to it. And once again, we're doing all different types of agents. We're doing CAR Ts, bispecifics, combinations, and now in context of the same pathways and same cytokines you've seen induced with COVID, asking those questions. And until we have a mouse model that's focused on humanized mouse model, where you have actually COVID infection and COVID directly induced CRS, our platform is a good way to get started until we have that perfect model.
Well, and I'm not going to ask you to go any further than that, because I know you've already hinted to me that you've got something in the works, and I'm very excited for when you are ready to start showing that off. I really am interested in the idea that folks are exploring inhibitors of CRS with us. Are they using these in the context of known controls for CRS, or are they simply dosing it with their compound of interest that they know causes CRS?
So in the case of inhibiting CRS, people are looking at our positive controls that are well validated. So all our PBMCs we use are leukopacs. So we can use essentially 2 billion cells that frozen away and go back to over and over and over again. We pre-screen them all for how they induce various positive controls are induced in those PBMCs in induction of cytokine release syndrome. People are looking at different drugs for different pathways. So different controls that you would want to use, which we want to use OKT3, or you want to use T Gennaro, or you want to use a super CD28 super agonist. There's lots of different ways you can induce it, and then looking for inhibitors. We have yet to have somebody come to us and say, I have a drug that causes CRS and I have a drug that inhibits it. Okay, but I'm sure that's going to come eventually.
So we have a question from the audience about what kind of major toxicities we're seeing from immunotherapies. I would say, in my personal experience, not a whole lot. To some degree, certainly we'll see mice, humanized mice, get sick on study. We're not necessarily verifying that sickness is indeed CRS. There have been some indications in some experimentation that we've done that at higher dosing there could be aggregates of the antibody mixtures that seems to cause a particularly adverse reaction, and backing off on that concentration seemed to prevent those aggregates and minimize that toxicity. Jim, can you comment on any toxicities that you've been seeing?
Yeah, I'm seeing something different, Aaron. So in our platform, what we're seeing is obviously cytokine induction. Okay, we see high levels of IL-6 or TNF, things like that. The onset of induction. Some combinations can have changed the pattern of induction of different cytokines. We look at clinical observation of the mouse. The mouse actually gets sick with some therapies. We could actually measure a scoring panel of 0 to 4. And look at immunophenotyping. The cytokines themselves can cause a loss of immune cells in the peripheral blood, like CD4s or CD8s. It's actually drug specific, which cell populations are actually depleted by that. We can actually, we do those kind of studies. And we've got some preliminary data looking at histology. We can actually see some organ damage downstream as well. It needs to be repeated and validated with more drugs, but we are seeing downstream organ damage, as well as looking at immune cell infiltration of organs as well. So essentially, we in body weight, obviously, things like that. So we're looking at a lot of different parameters, and not every drug is affecting all these different parameters. But essentially, we're seeing this sort of toxicity on the mouse that's drug related. It seems to be correlating with cytokine release.
That was perfect. I think that was a great note, actually, to sort of start to end this, because we're right up near the end of our time. I'm going to thank you, Jim, for your time today. I really appreciate the conversation. I'm pretty excited about the new platform. And I think what I'd like to leave the audience with today, if you're interested in following up on anything that we discussed, is that you can easily reach out to us through our customer service, or you'll see the email below, mystech@jax.org. That reaches out to our technical information scientist team. They will get you in touch with the right folks to have some further conversation on the topic. And ultimately, if you want to run CRS with Jim's team, get in touch and we'll make that happen. Typically we do these every week, but we're skipping next week. So join us in 2 weeks and have a nice 4th of July holiday. Thanks again for joining us. And thank you again, Jim, for your time today.
My pleasure.